Sushruta for NEET is now available on the Google Play Store. Try it out for free.
Good morning, everybody. Let us study now question number 61. Quick. Janani?
Sir, D.
Option?
Deer.
Dog. 62?
B.
Huh? Hello? What is the answer?
So for the 61, it's D, no, sir?
It is B. Phosphodiester bonds. 63?
A.
A. 64? Why so much time?
C.
C. 65?
A.
Yes. 66?
B.
B. DNA. 67?
C.
67?
C.
Who is telling this?
Plexi, sir.
Sharon?
Sir, deer.
67. Answer is 20. Why?
Because they have not asked the base pair.
How many nucleotides are present in one turn of DNA helix? 10 base pairs. How many nucleotides in each base pair?
2.
2 into 10, 20. 68? 68.
A.
Yeah. Please note down there. You know what all different types of DNA are there? Aisha? Aisha? Kenisa? Do you know what type of DNA are there? Janani?
No, sir.
Kavita?
No, sir.
Please note down. There are, you just write down one by one below the another. There are what is called aDNA, can you see this board?
Yes, sir.
Yeah. Here what is called aDNA, bDNA, cDNA, ddNA, eDNA, right? zDNA. All these DNAs, right from A to E, all these DNAs are right-handed. They are all right-handed DNA. Okay. Please note this bDNA is Watson and Crick DNA. Watson and Crick DNA is bDNA. And these have different arrangement of base pairs. A has 11 pairs, B has 10, C has 9, D has 8. Okay. ZDNA has a 12. ZDNA is left-handed. Left-handed. Okay. Just this information, you please have it. Right? Next. Let us go to 68 question. What is the type of coiling in DNA? Whenever we ask DNA, it is Watson and Crick DNA. It is right-handed. 69. Yake, don't waste my time. Blessy,
Yes, sir.
what is answer?
I think D, sir.
Huh?
D.
Yeah. So 69 is helicase. Helicase is the one which is responsible to separate two strands of DNA. Helicase. Then 70. Jyoti, please answer that question. Jyoti, are you there? Rafa? Rafa? Rafa, are you there?
Yes, sir.
What is the answer? 70. A double-stranded virus DNA with 20,000 base pairs in nucleotides.
40,000.
Yeah, it is 40,000. Dear. Because each base pair has 2 nucleotide, it is 40. 71. Janani. Janani. Are you there?
Yes, sir.
Tell me answer quickly.
U
What?
GUCTU. No.
How can it be?
GUC.
When it is C here on DNA template, what should be on mRNA?
G.
G. In the place of T,
U.
U. In the place of G,
C.
C. So we have here mRNA sequence. In the place of CG,
Option A.
option A.
Sir, option B, right?
Option A is correct.
Option B, sir.
It's option B, sir.
Sir, option
Option
B.
B. Why? C means
I am T means C. A.
Sir, T.
G. T
for-
means A. Is it A or U?
A, sir. In place of
A.
thymine, we
A.
put.
A.
Yeah,
A.
sir.
So answer is B and not A. Understood? Janani.
Sir.
Yeah. 72. Namrata.
A.
A. What is it codes for?
Methionine.
Methionine. And it is also third codon. 73.
C.
73C exons. 74. Sarvotam.
Yes,
Sarvotam.
sir. Yes, sir.
74.
74, sir.
Yeah.
C, sir. mRNA.
Simply you are telling.
No, sir.
Anyone else?
D.
Single stranded RNA present in the following bacteria. What is it? TMV?
Tobacco mosaic
Tobacco mosaic
virus.
virus. mRNA. What is the answer? Navya?
All of these.
All of these. 75.
C.
Aisha?
C.
Aisha? Are you there?
Yes, sir.
Quick. 75. Kanisa? Kanisa?
C.
C. What they are called? What is your
Stop
answer?
codon.
Top codons. 76. Prithviraj.
B, sir.
C. 76. UAC. 77. Vishwas. Vishwas. 77. Vishwas. Are you there? Are you there? Harshavardhan. 77.
C, sir.
C. Carry amino acid to ribosome. 78. Vispita. Vispita.
Yes, sir.
78. Why so much time? Chinmayi. Pardon? C. mRNA. 79. Kavita. Kavita.
Yes, sir.
79.
C.
C. 80. Siddesh.
DNA.
Siddesh.
C.
One of the following is not
C, sir.
directly involved in translation. Charan. Nadira.
Yes, sir.
What is the answer? Question number 80. Sarottam. Blessy.
C, sir.
C. DNA. 81.
A.
81. A. 82. Diti. Diti, are you there? Diti. Janani. 82. Janani. It is in virus. Don't bother about what virus it is. Which of the
B,
following
sir.
sequences found in virus?
C.
B. RNA to DNA to RNA to protein. In all viruses, it's a retrovirus, you know. Okay. 83.
B.
Namrata. 83B. 84. Namrata.
A, sir.
A. 85. Navya.
A, sir.
A. 86. Rakesh.
A, sir.
A. 87. Aisha. Aisha. Aisha, are you there? Kanisa. Kanisa.
mRNA.
mRNA. In mammals, it lives for few minutes to 2 days. In bacteria, they live from seconds to an hour. In most of the organism, it lives within 1 to 3 minutes. 88.
C.
B.
88. Prithviraj.
C, sir.
C. 61. Vishwas. 89. Vishwas. Are you there?
Yes, sir.
Helapa. 89. Which of the following sequence code for 4 pyrimidines? Which are the pyrimidines?
Cytosine.
Thiamine.
Cytosine.
Cytosine and
Cytosine.
thiamine.
Cytosine and thymine. Adenine and guanine.
Urine.
Yeah. Here question is, which of the following sequences code for 4 pyrimidines? Where you have 4 pyrimidines? In which of the group?
C.
C. Answer is 89C. 90. Harshvardhan, 90. Harshvardhan, are you there?
Yes, sir.
What is the answer?
Translation.
90.
Translation, sir. B.
Translation B. Is there anyone whose name I have not called so far? Hello? Hello? You don't want to say? Whose name I did not call so far today? 91? Vismita?
Yes, sir.
You know? You know answer for 91? Chinmay?
C, sir.
What is the answer?
C. It has a
Loud.
like structure.
Huh?
C on
Which,
C option.
which of the following characters is not applicable to tRNA? It is the smallest of the RNAs, correct. It is made up of 75 to 95 nucleotides. It is formed of 75 to 95. You know, tRNA is also called Janani. Navya?
Adapter.
Huh?
Adapter.
Namrata?
Soluble RNA.
Soluble RNA. Yes, RNA. So, it is the smallest of the RNA. Right? It is correct. It acts as an adapter for amino acid. Yes, it correct. It has a clover-leaf-like structure. Correct. It is the largest of the RNAs. This is wrong. It is wrong. It is not the largest of the RNA. Largest of RNA, which one? mRNA. mRNA is the largest of the RNA. 92. Kavita? 92. What is the answer?
A.
A. RNA polymerase. Siddesh? 93. Siddesh? 93. Charan?
Sir, B.
Yeah, 93.
B, sir.
B. A.
No, A, sir.
in
A.
trans- A is here A. What should be there?
U.
U. So this A.
U.
U. That means
Option D.
option D. 94.
C.
94 is a C. DNA and RNA different, base and sugar. 95. Which is correctly matched. Nadira? Nadira?
C, sir.
C. 96. Sarottam? Sarottam?
Sir, I
What
think
is
it is
Exon segments are reunited after splicing by Blessy? Blessy?
C, sir. C.
Pardon?
A, sir.
What simply you are telling?
D.
Reunited. Reuniting is requiring
D,
which
sir.
enzyme?
D.
D, sir.
Ligase. RNA ligase. 97.
B.
97. Diti, Rafa,
Yes, sir.
97.
Is it B, sir?
B, promoter. 98. Namrata,
Sir,
it is? It is? Degeneration of genetic code is due to third member of the codon. Third member of the codon. You see here? Now, look into this. You have here AUU. Can you tell me for which amino acid it codes for? Come on. You have to by heart, I said. For which amino acid it codes for?
methionine.
Huh? Isoleucine. It is for isoleucine. Is it not that these are common? Which one is changing? Third member of codon. Did you understand this point, Navya?
Yes, sir.
Okay. So like that, if at all you see, what is this? AUG. This also same. But this is different. All these codes for isoleucine. These codes for
Methionine.
methionine. Methionine. Okay. Answer for question 98 is a C. 99? 99. Pratyuraj.
C, sir.
C. Okay, sorry. Right. 100. 100. Harshvardhan. Kavita.
Yes, sir.
100.
C.
Hmm?
No, sorry. B.
Portion of gene which is transcribed but not translated. Transcribed means it gets formed, but not translated means it is not coding for any protein. Answer is B. Okay, open now. Start answering. 101. Anyone? Which one is not applicable in respect of genetic code?
Overlapping.
Overlapping is which one is not applicable in respect of genetic code? Overlapping is it not that you would come across? Some of these amino acids coding for the same, some of these codons coding for the same amino acid. What is not applicable is
B.
redundancy. Redundancy. That means no longer needed. There is no amino acid which is said to be no longer needed, not useful. Out of 64, all are useful. 1 or 2.
B.
Huh?
B.
How? Which enzyme will be produced in a cell in which there is a nonsense mutation in the lac Y gene? It is A. Right? 103.
A.
A.
A. 104.
A.
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 105.
A.
A. 6.
A.
6.
D. D.
Dog. Translation, transcription. 107.
Sir, can you scroll it down?
Hmm?
Can you scroll it down?
107. Why this much of time?
B.
B. Amino acids. 108.
C.
C. 109.
D.
D. 110.
A.
Amino acid sequence in
C.
protein synthesis is decided by
C.
C.
mRNA. C. 11.
A.
A. Is it that a single gene is formed of only one triple codon? Only 3 bases?
No.
Then
D. D, sir.
D. A single gene is composed of part of a strand of DNA double helix. Is it not? It can be, I have mentioned yesterday, largest gene. You will be having sometimes several thousands. Okay. 112.
B.
B. And
B.
13.
C.
Pardon.
A.
C.
Simply you are telling. Who said A?
Me, sir.
Who said A?
Navya.
C.
Navya. Navya.
Yes, sir.
Is it UAA, UAG, UGR starting codons?
Oh, no, sorry, sir.
Mm,
Just stop
you-
codons.
So, UAA, UAG, UGA are nonsense codons. What do you mean by nonsense codon?
They're the stop codons.
Top codon. A nonsense codon is one, a codon for which no normal tRNA exists. Please take it down. A nonsense codon is the one for which no normal tRNA exists. So when that codon is there, when the protein synthesis machinery passes nearby that, protein synthesis stops. Because in that region, no fresh tRNA comes and offers its amino acid to the growing chain in the P site. 114.
D.
D.
D. Please note down there, these UAG, UGA, UAA are called by a certain name. UAG is also called amber code, amber, AMBER. UGA is called OPAL, OPAL. And UAA is called ochre, OCHRE, OCHRE. I hope you have understood. Did you take down? You have 3 terminologies. UAG is called what? Amber. Amber code. UGA,
Okay.
Opal. And UAA, occur. Okay? Right. 115.
A.
115 is A. Is it? The activation of amino acid during protein synthesis requires ATP and enzyme synthetase to attach that amino acid to the tail part. 116.
A.
A. 117.
A.
A.
A. 118.
C.
The site of mRNA which binds it to tRNA molecule is site of the mRNA which binds it to tRNA molecule is none of
A.
these. None of these. Why? Why? The site of mRNA which binds it to tRNA molecule is. Prior to that, you come across. You don't know. Okay. You think over this question. Why that? The site of mRNA which binds it to tRNA molecule is. I am giving another question. 2 questions I have given. On the other day, I have given, and today another question. 119. Hello?
Sir, can you scroll?
Hello?
C.
Quick, quick, quick. It is already 6:40.
C.
Huh?
D.
Pardon?
D.
D. All of these. 120.
C.
C.
C. 21.
B.
B. mRNA. 22. Cistron. 23.
D.
D. Dog. 24. With adenine and cytosine only, how many different codons are possible?
A.
How? Who said it? Who said it?
Canisa.
Correct it is. How?
Because in adenine 4 codons are possible and cytosine 4. So 4 plus 4, 8.
Yeah. In adenine, 4 codons are possible. What are they? In adenine, you said 4 codons are possible. What are they? Come on, Kanisa. Adenine, 4 are possible. You said 4 are possible. What are they?
A, C,
A,
A,
A, C,
A, U,
A, C, C,
A, U, U,
A, C, A. You, how you can bring a U? What is the question?
with
With
adenine and cytosine only.
adenine and cytosine only. Am I right? Adenine and cytosine. Sorry, this last one I made mistake. What is that? Correct?
Yes, sir.
Correct? So here, with the C, correct? Correct? So how many? 4. 4 plus 4. Did you catch my point?
Yes, sir.
Only 2 codons are used here. Okay? 125. The genetic code was discovered by Nirenberg and Mathai. 126.
20.
The total number of essential amino acid. I think you have not taught. I shall take up this in some other context. I think I have told you. There are essential amino acids, non-essential amino acids, conditional essential amino acids. So the total number of essential amino acids in the living being is 8. The answer dog. 127. The first mRNA synthesizer for the assignment of codon was polyuridylate. This one. Answer dog. 128.
A.
A. Degeneracy.
A.
Degeneracy. 129.
B.
AUG.
AUG. 130.
6.
6. Can you tell me what are they? CGU, CGC, CGA, CGG, AGA, AGG. 6. 131. It is very, very important. You must by heart this genetic code. Remember. 131.
C.
C. 32.
A.
32.
C.
C, sir. C.
33. How many
A.
code for the amino acid serine?
A.
A. 6. Here also again. UCU, UCC, UCA, UCG, AGU, AGC. 134.
B.
B. The code is a triplet. 35.
A.
A. RNA to proteins. 36.
C.
C. 37. Rakesh. Rakesh.
Yes, sir.
Yake, why you are so keeping quiet? 137. What does lac refer to?
A.
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 Lactose. 138.
C.
C. Lactose dehydrogenase. 139. E. coli cells with a
A.
mutated Z gene. Yeah?
A.
A. Z gene is concerned in the beta galactosidase. 140. Lac operon is an example of
C.
inducible operon. 141. In inducible operon, regulatory gene releases
C.
C. Repressor. 142. Operon system consists of C. Chromophore and all of the above. 143.
D. D.
B.
Dog. 144.
D.
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 Dog. 145.
B.
B.
B. 146. What? ESTs are 146. ESTs are genes that are expressed as
A.
RNA. B. 147. Steps in a sequencing human genome project. Which should be the first?
Isolation.
Option B.
Option
B.
B. 148.
B.
B. 149.
D.
D.
B.
May 2006. 150.
C.
Is
A.
it?
A.
A. Very important. 151.
2.
Yeah?
Option A.
A. 151. 151. A. 152.
C.
C.
53.
2%.
2%.
A. 54.
D.
D.
Dog.
D.
55.
B.
B. Non-coding function. 56.
A.
A. 57.
A.
No.
C.
C. SNP. Single nucleotide polymerase is location of genome where the single base of DNA differs. 58. A.
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 59.
B.
B.
Yes, dog. 60.
B.
1 minute.
B.
50. 59. 1 minute. I made a mistake. 58 in human. Yeah, 58 correct. 59. Oh, there is correction. Here, this is 59. Oh, one BAC, this is 59. You put a question mark here, 59, 159. BAC and AYACs are dog. This is not 59. You consider 160. I will correct it and put again. 160.
B.
B. Alex Jeffrey. Next question. V and TR is employed.
B.
B.
B. DNA fingerprinting. Next. DNA fingerprinting is related to molecular analysis of profiles of DNA sample. Next. Here. Which of the following pairs of term name means one and the same things?
D.
D. DNA fingerprinting means DNA profiling. Yes. Next, DNA fingerprinting refers to
D.
yeah, D is correct. Analysis of ENTR of DNA sample. 64 here. Next. Southern blotting technique. Isolation of DNA from a nucleated cell. Next. Which step does not involve the DNA fingerprinting?
D.
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 Yeah. It is related to
RNA.
RNA. Only that much. Don't get confused about it. There is no much about that. Next question. Here 166.
C.
B. Southern blotting. Next 167. DNA probe is used for
A.
it is all of the above. Next question 169 and 68 here.
B.
Huh?
B.
Option B.
B. B. Next 169.
A.
A. $170. Sorry.
A.
Balcony. It's okay. 170.
D.
I said this is okay. 99. 169 over, bulk of DNA. Scattered DNA. This one, scattered DNA or repeated DNA. Answer is 170.
D.
All of the above. Correct. 171. Basis of DNA fingerprinting is. What is that?
A.
A. 172.
C.
C. Variable number of tandem repeats. 173. The sensitivity of DNA fingerprinting can be increased by using polymerase chain reaction. That means it goes on doubling. We are going to study, have not studied this PCR. Once you study PCR, it becomes very clear to you. Yes, 174.
D.
Yes, correct. 175. Which one represents the correct manner of DNA replication? You must know what is the lagging
C.
strand. Pardon?
C.
Pardon? You must know where you have 5 prime, 3 prime, in which strand you have, which is the coding strand, which is the template strand, lagging strand, occurs the key fragments. Which one holds good for this?
A.
Is it A? You please look into all these.
C.
C,
Answer
sir.
is D. Dog. Please look into that. In your book, compare with that. 176.
Sir, C and D are same.
Is it?
Yes, sir.
1 minute. Okay. Okay. Right. Okay. So 176.
D.
B,
No, sir, D.
dog. Okay. 177. One 177. Why so much time? I have told you, you must know which particular experiment is related to which individual.
C. Option C.
Option C. Right? Okay. 178.
C.
Yes.
C.
C.
179. Why so much time? What is the doubt? Hello?
A, sir.
How? Simply don't tell. See, this is how much? This is starting. This is how much? What proportion here?
50-50.
Huh?
50-50.
50-50. What should be here?
25-25.
50-50. 150 should increase. Another 50 should decrease.
75-25.
A heavy DNA of equal is allowed to replicate in N4 medium for 80 minutes. This is first 20. This is 40. Then 60. Then 80. Okay. 70-25. When this is 50-50 when? After 20 minutes. After 40 minutes, what will happen? 75-25. After another 20 minutes, what will happen? Hello? See, in the first 20 minutes, it doubles. Plus 20. This is how much? 50-50. Next one, 50 increases, another 50 decreases. How much it comes? 75-25. Next, what will happen? Not catching my point. Not catching my point.
So one of them will decrease.
Yeah, this holds good for what? To this, am I right?
Yes.
Yes.
50-50.
Yes, sir.
In the next step, what should happen? This should come. Am I right?
Yes.
Right? This is
Yes.
to which one?
Generation 2.
That means Is it this one?
Yes, sir.
Right? 20, 50, 50. And after 20 minutes, this should be seen in only one stand. Yeah, one stand. This is 25. And this will be 75. Then what will happen? What will decrease and what will increase? Find it difficult? Hello?
Yes, sir.
Find it difficult? Why?
Light
Now.
will increase.
Pardon?
Light will increase.
Light will
That
increase. Light will increase. Out of this, which one you have to select?
C or D?
D, sir.
D? Is it
C,
all
sir.
hybrid DNA?
No.
Then
C, sir.
C.
C.
C is answer. Am I right? Next. 180. What is the answer?
C.
C.
A.
A? The silent observations done from Human Genome Project. Choose the correct option. How many nucleotide base?
C, sir.
Yeah? C is the answer. 181. Unless you know, you cannot. Can you answer the question by seeing the textbook?
B.
B, correct. 182. Hello?
A.
Is it? What is this tRNA concerned with? Of course, this 1, 2, 3 should be,
Anticodon.
yeah? tRNA concerned with
Anticodon.
anticodon. tRNA. Anticodon. Then, so is it there anywhere? tRNA. Anywhere? Okay. There is no one tRNA anticodon. Then, anything else? Which is the initiation codon?
Sir, C, sir.
AUG.
Yeah?
C. Third one. C option.
Option C. Correct?
Yes, sir.
How?
AUG initiation codon.
ALB initiation codon. That is 2. That is 4 is 4 is
Lagging strand.
no. Is it that you said C? No. Who said C?
Yes, sir. Kavita.
Is it correct?
Yes, sir.
Is it perfectly matches? Or is there mistake?
Wait, sir. I'll see.
Huh?
So the numbering is a bit different.
Wait, sir. I'll check.
Is there no mistake? You don't see this number. You don't see ABCD. You just start marking on this. Can you mark? Near A, what you can mark out of this 1, 2, 3, 4, 5?
Fourth.
You
Fourth
mark
one.
A, 4. Then B,
Fifth one.
Fifth one.
huh?
Fifth one.
Fifth one.
Okay. Then C,
First.
First.
first one. D,
Second one.
second one. E, third one. Whether you have that matching here, correct matching?
Yeah.
Do you have correct matching here? No. Okay. Right? You don't have. There is a mistake in this. Yes, come on. Next question. What is the answer? Sorry, what is the answer?
C.
C. Next question. What is the answer?
C.
C. Next question. What is the answer? Study the following molecules related with their anticodon. For that, you should know what is the codon anticodon. What is, which type of RNA tRNA is impossible? T1, 2, 3, 4, 5. Can you read that codon? Anticodon? Yes or no?
Yes, sir.
Yeah, tell me then what is impossible? What is impossible? Look into the codon, anticodon, and decide. Next. I have left three questions to you. Let me say that who will give the answer. Next one. Can you see this? What is ABCD stands for? You know where is that A?
A
What
is
is
RNA
it?
polymerase.
Yeah.
B is sigma factor.
Okay. So answer is
A.
A.
A. 187. All these are figure-based questions. 187.
D.
Dog or B, bat? Hello?
B. B. B.
B, bat. 188.
C.
C, 189.
C.
C. I will do one thing. I shall put the answer, this MCQs with the correct answer boldened in Google Classroom. You see there? You have this also without answer, and I'll be uploading with the correct answer bolded with regard to each and every question. Okay? Right. Now, any doubt you have? Any doubt you have? Yes?
No, sir.
You don't have any doubt. I would like to take up new chapter, that is human health and disease. If at all we complete this chapter, only the unit 1 chapter will remain, and I will take up human reproduction after finishing this one in my backlog. Right? So we shall take up this human health and disease. Are you seeing the board?
Yes, sir.
Yeah. Biology is the youngest of the formalized disciplines of natural science. It is the most recent one as far as the research is concerned, or advancement of research is concerned. Progress in physics and chemistry proceeded much faster than in biology. Applications of physics and chemistry in our day-to-day life also have a higher visibility than those of biology. That is why there is a separate chapter in chemistry. What is that? Chemistry in daily life. However, 20th century and certainly 21st century has demonstrated the utility of biological knowledge in furthering human welfare, be it in the field of health sector or agriculture. The discovery of antibiotics and synthetic plant-derived drugs, anesthetics, have changed medical practice on one end and human health on the other end. So you know how much important it is in the field of medicine, anesthesia, right? Life expectancy of human beings have dramatically changed over the years. Now you see lot of old-aged people still healthy, doing their work on their own. Agriculture practices, food processing, and diagnostics have brought to socio-cultural changes in human communities. These are briefly described in this particular chapter. And you must know this particular person, who is this? Swaminathan. Right? Why you should know him? He was responsible in developing short duration, high yielding varieties of rice, including your scented basmati. And he is also worked with Norman Borlaug, and his work culminated in the Green Revolution through introduction of Mexican varieties of wheat in India. Therefore, he is credited with regard to the Green Revolution also. Now let us take up this particular chapter. Health for a long time was considered as a state of body and mind. This is very important, body and mind, where there is a balance of certain humors, certain fluids. This is what early Greeks, like Hippocrates, as well as Indian Ayurveda system of medicine asserted. It was thought that persons with black bile, one of the four humors of ancient and medieval physiology that is believed to be secreted by kidneys and spleen, black bile, we don't agree with regard to this, and to cause melancholy. Melancholy is some sort of depression, sadness when present in excess, belong to hot personality and would have fevers. For a very long time, it was considered that it is black bile that is responsible for all this depression and having fevers. But we rule it out now. This idea was arrived at a purely reflective thought. The discovery of blood circulation by William Harvey. It's very important, always ask a question, who discovered blood circulation? William Harvey, using experimental method and a demonstration of normal body temperature in person with black bile. Normal body temperature, using the thermometer, disproved the good humor hypothesis of health. Nothing like that, good humor, bad humor, good bile, white bile, black bile, like that. No. William Harvey has made it very clear that there is nothing like fluid, there is nothing, any association between black bile and the fever. In the later years, biology stated that mind, this is very important, mind influences through central nervous system and endocrine system, our immune system, and that our immune system maintains our health. That you agree. If at all you have very good health, means your immune system is so strong. Your immune system becomes so strong because of the mind influencing the neural system and the endocrine system. But if at all you decide to open the brain and see where is the mind, you will not get a mind like that. Again, it's a chemical. So mind influences through the neural system and endocrine system, our immune system. If at all you want to be healthy, you should have very good immune system. That means with good resistance power. Hence, mind and mental state can affect our health. So always mind or our mental state affect our health. If you feel that, yes, I have eaten ice cream today, I am going to get cold tomorrow, you will definitely get the cold tomorrow. It is your mind that is important. If you feel that, yeah, nothing happens to me, I am very strong, always you must tell, I am very strong, I am very strong. You will become strong because mind influences your health. Our health is affected by, of course, genetic disorder. There are certain disorders which we get right from the birth. Genetic disorder, deficiencies with which a child is born. Deficiencies and defects which the child inherits from parents from birth. It is a genetic disorder. Genetic disorders are the disorders which we get right from the birth, which we get from our parents. That is one type of problem they got health. The another one, the health is affected by infection. That is true. Many times whenever you go out, many times whenever we eat out, we may get infected. And this infection is possible by different agents. And more than that, our health is affected by lifestyle, including food and water we take, rest and exercise we give to our body, habits that we have and lack. This is very important in deciding your health. Remember, this chapter, human health and diseases, don't study with the view of only examination. Please study in view of your good health in future. The lifestyle, including food and water we take, and we have the habit of eating whatever that is available, wherever available. Is it correct on our part to eat the food items which are kept on the roadside, open, without any coverage, and enjoy eating such food? Navya? Navya?
Yes, sir.
Is it good for you?
No, sir.
Then why you are eating?
I don't know, sir.
You don't know.
Not, sir.
Kanisa? You don't eat outside?
Yes, sir.
Is it correct on your part? Is it proper
No,
on
sir.
our part to eat the roadside sold food? Pratyaraj? Namrata? Jyoti? Siddesh? What? Tell me. Is it not that we are least bothered about our health because we eat food items not at all maintained cleanly with hygienic condition? Pratyaraj,
Yes, sir.
are you not eating pani puri at the roadside?
No, sir.
Then, so the lifestyle including food and water we take, rest and exercise. How much rest and exercise we give to our body? Of course, you may take rest now. But are you giving exercise to the body? You think exercise is for Buddha Buddhis, aged people, and not for you. Habits that we have and like it. Good habits that we have, or bad habits that we have, or good habit we lack. What is that good habit we lack? Going a walk early in the morning is a good habit, but we don't want. Doing a little bit of exercise is a good habit, but we don't want. Cigarette smoking is a good habit, am I? No, bad habit that we have. Whatever good thing that we want, we like it. Whatever we don't want, our body doesn't want, we have it. So this is very important in your governing your health. Take care of your body yourself. The term health is very frequently used by everybody. How do we define health? Many times we say, I am healthy because I don't have any disease. Health does not simply mean absence of disease or physical fitness. I am physical fit. I am physical fit. I am healthy. Don't say that. Don't say. You can't make it out. It could be defined as state of complete physical, mental, and social well-being. If at all you want to say that, if at all you want to claim that, yes, I am healthy, you should be physically healthy, you should be mentally healthy, you should show social healthiness, right? When people are healthy, what is the advantage? They are more efficient at work. If at all you are healthy, you need not bother about the health issues because you are healthy. You don't have any issues at all with your health. That is most important right from now till you live your full lifespan. I bless you. I pray God that you please all of you should live your full lifespan crossing 100. This increases productivity. When you are efficient at work, increases productivity and brings economic prosperity. Health also increases the longevity of people. They live long on this earth and reduces infant and maternal mortality. Infant mortality is the death of the young babies. Maternal mortality is the death of the mother. It reduces. Health reduces the infant death, mortality, and maternal mortality. Balanced diet, personal hygiene, and regular exercise are very important to maintain good health. So, whatever you want to forget, you forget. I don't mind. But please don't forget these sentences for your own welfare, for your own good. You should have balanced diet. You should have personal hygiene and you should have regular exercise. And these are very, very important to maintain good health. I hope you know what is balanced diet. Balanced diet includes eating very much fried item, eating ice cream, eating sweets, eating chocolates. Am I right, Navya? Navya,
Yes,
am I
sir.
right? Yeah?
No, sir.
Then
It's
you
all the
should have balanced
nutrients
diet.
in a diet.
And you must start your diet eating with vegetables. With vegetables. You must start eating fruits with empty stomach. Eat vegetables. Fill your stomach half with vegetables. Then go for that. But what we do? We start our lunch or dinner sometimes with the starter. Then we go for appetizer. Am I right? Am I right? And with this starter and appetizer, about half of the stomach gets full, filled. Then we go for again some more fried items. Then all the sweets and all. When it is required food, and then at that time there is no space in your stomach because they're all full with this. So you should eat more of vegetables, fruits, and then you have to then go for and what you should minimize. You should minimize eating sugar rich food and oil rich food. Yoga has been practiced since time immemorial to achieve physical and mental health, which is a yoga day, International Yoga Day,
21st June.
21st June. Awareness about diseases and their effect on different bodily function, vaccination against infectious diseases, proper disposal of waste, control factors, maintenance of hygienic food and water resources are necessary for achieving good health. Do you know what all are necessary to achieve good health? When the functioning of one or more organ or system of the body is adversely affected, characterized by various signs and symptoms, we say that we are not healthy, we have a disease. So, if at all we have it, if at all we want to know that we have a disease, there should be some symptoms of the disease. Unless there is symptom of disease, we consider that we are healthy. Nothing is happening to me. I am very healthy, I am very strong, I am healthy. But it need not be. It need not be. So, diseases can be broadly grouped into infectious disease, and non-infectious disease. Diseases which are easily transmitted from one person to another are called infectious diseases. Infectious diseases are very common, and every one of us suffers from these at some times or the other. Some of the infectious diseases like AIDS are very dangerous, fatal, life-taking. Among non-infectious diseases, cancer is the major cause of death, non-infectious. AIDS is infectious disease. Drug and alcohol abuse are also affect our health adversely, right? So, I am going to take up these common diseases in humans in my evening class. Okay? Right? Thank you. Bye. See you. Have a good day. Any doubt you have?
Thank you, sir.
No.
No, sir.
Okay, bye. See you.